Welcome to week 3. Okay, so this week that on Monday you've got your exam, so that'll be due on Wednesday, and we talked about that in the last video. Then the other two things we're going to do this week is going to be involve DNA. So we hear that DNA is the information of life, and that holds all of this information. Well, we're going to look a little bit into the next couple of chapters into how exactly that works. And so you'll be looking at DNA, what it is, how it functions, and how it's replicated as we go through producing, because you start out as a single cell and then now you're billions of cells. So how do we make sure each cell has the exact same set of information? Well, that's through this replication process. And so you look at DNA replication, and it's kind of in a cool way. This is what actually sold me on this textbook in the beginning, is this idea of using DNA in forensics. And so you're going to get some insight into how DNA is used in forensics, which is really just taking advantage of what happens in nature, of how DNA replicates itself in nature. And it's something that we do a lot in science, when a lot of science research involves manipulating stuff that cells do anyway. It's the natural process, and we just figure out how to tweak it to work in our favor in terms of doing research. So you'll be looking a little bit at forensics, at how forensics are used and how DNA is replicated in that process. So you'll look at DNA replication, and then the other chapter that you're going to look at is how do we get from the information to actually what you have in terms of an organism. The same sort of way you get from a computer code to the actual program. We have to do the same thing with DNA. We have to find out it's coded information in A's, T's, C's, and G's, but how does that translate into who you are, the hair color you have, the eye color you have, your height, your ability to process sugars. How does that work? Well, we'll look at how DNA leads to proteins and how that happens. And so Monday you won't have a lab or activity because you have that exam that you're working on for Wednesday. And then on Wednesday, you're going to be doing a lab activity involving DNA and isolating DNA out from a piece of fruit. And recommendation is either strawberries, bananas. You can try whatever. People have done onions before. You may not want to smell up your house, but onions work pretty well as well. Strawberries, bananas, and onions. I've seen kiwi. You can try anything that you want to. There'll be some that'll work better than others. It's another great activity to get children involved in if you have kids around that want to help. This is a cool experience. I'm actually considering doing this lab this summer with my 2 girls who will be at that time 4 and 7. And I think it'd be a lot of fun to do this lab with them. So it's a fun lab to do, isolating DNA out of pieces of fruit. And you get to see this cool, snotty DNA. And then the second thing you're going to do this week on Friday, that'll be due on Monday, is taking DNA and taking it through the RNA step and then into proteins, and looking at how mutations or single changes in the DNA can really affect what that protein is at the end. And sometimes it affects it in a bad way, sometimes it doesn't affect it at all, sometimes it might be a good effect. But it can have an impact if we change the DNA, and that's going to be important as we move forward and start talking about cancer in a little while, is how those changes in the DNA result in altered functions in the proteins that are being made. So you're going to be doing an activity. I highly recommend that you look at the video. At the beginning of each of these labs and exercises, you have a read this, watch this. Highly recommend that you look at the video. I made it last summer and walked through the process of transcription and translation of going from DNA to RNA to protein. And I think that will be helpful as you do this lab. We had some students get confused last summer, and it turned out that they hadn't watched the video. So hopefully, if you watch the video and then look at the example that's given you in the lab as to how to start this, I think it will go smoothly. It's going to make you go a little bit cross-eyed because you're going to be looking at A's, T's, C's, and G's. And I would recommend splitting them up into 3s because that's how you're going to read them for the protein. So do 3, put a space, and then 3, space, and then 3 again. I think it'll be a little bit easier to follow if you do it that way, and a little less conundrum in your brain. All right, and that's it for this week, and I look forward to heading into week 4 in our midterm week that we're already halfway through. All right, bye.